Thank you all for coming out tonight. We have a really outstanding speaker, Dr. Brian Bennett. Brian joined us about a year and a few months ago, and he came to us from UCLA, where he was doing his postdoctoral research work, and before that got his Ph.D. from the University of Washington. I guess that's the one in Seattle, not in St. Louis. He works in the field of genetics and how our genes influence whether we get heart disease or not. Probably 1 in 4 of the people in this room are going to die of heart disease. It's one of the big chronic diseases that we would love to stop. Clearly, nutrition has a big role in it, and clearly there's something about our genes that interact with nutrition that place us at risk. And Brian's skill has been to use animal, rat models, mouse models, and to use those mice to try to uncover what those gene, nutrient, and heart disease links are. And his talk tonight really will talk to you about some of this research he's doing. He's been awarded a grant from the NIH to study this, and some of what you'll learn about today is how do we figure out which of our genes and which of the nutrients we eat are placing it at more or less risk for developing heart disease. So I welcome Brian Bennett. You'll enjoy the talk.
Cool. So I think you guys can hear me, and we'll just get that out of there so we don't have any feedback problems. Well, thank you all for coming. I really appreciate it. It's quite an honor to give a talk like this, and I can see. I'll probably give the talk from about here, so I think I can see both sides. All right. So I've given a couple talks in Kannapolis for non-scientists, and they're actually one of the harder things to do. And I have to think about all the things that probably I learned along the way that I need to explain in a way that makes a lot of sense. So when I stop making sense, whoever's not bashful, you can start to raise your hand, and I might slow down for a second. I won't think you're rude. I'd like you to get something out of this. So keep me honest that way. And every time I give one of these talks, I get an important question. And it's a really important question if you're from around here. And it's this one. Who is your favorite driver? I've had that as the number one question, so I've gotten done the whole talk. This is the most important thing. This is my life's work. And that is actually a question I've gotten more than once. And it's okay. I don't take offense to it, because what I've decided is often people are thinking about this, but as a scientist, often I'm thinking about this particular person, because that person that drives those trucks brings me the things that I need to do my research. And so they actually stop by every day, and they're real important, actually. So coronary artery disease it has complex biology, and I have a list here of things that can affect your risk of coronary artery disease. Obviously, from a genetic side, oh no. Yeah. Sorry. Yeah, there we go. From a genetic standpoint, you have your family history, and then also there's genes that affect your LDL or bad cholesterol, your HDL levels, which are your good cholesterol, and a number of other things that you're going to go to your doctor to get checkups for to make sure you don't have, such as hypertension, diabetes, obesity. And obviously down here, age. Age is one of the most important factors that predisposes people to getting coronary artery disease. It takes such a long time to develop. And there's also environmental factors, such as having a high-fat diet, smoking, lack of exercise, perhaps some pollutants. And those are things that we wind up studying at the NRI. And so this is just another cartoon to show that there are diet causes and genetic causes of coronary artery disease, and they often flow through plasma lipids. So by lipids, your LDL, HDL levels. And in particular, when you have increased LDL and reduced HDL, you're going to be more susceptible to cardiovascular disease. And so in particular, what I'm interested in is a process called atherosclerosis, and that's kind of listed here. And it's a cause, an underlying cause of coronary artery disease. And so what I think about is personal risk. So often if you're here, you don't necessarily, and when you're in the doctor's office, you don't care what the statistics for coronary artery disease. You care, what's my personal risk? And that's not so well worked out. That's one of the things where we're headed in science, is understanding who's susceptible, in particular, to certain things that are going to make them more susceptible to coronary artery disease. And obviously, there are really good examples of people who are really resistant to coronary artery disease, and all the risk factors that you're not supposed to do, you can live to 100. And there are other folks that are just a little bit more susceptible. And these are just two prominent examples from the media of people who have either been susceptible or resistant to coronary artery disease. And so one thing about scientists, we really like data, and we like to present data. And so I'm going to just go through some slides giving some basic statistics about cardiovascular disease. And what's important about that is just to build the case that it is important, and what are some of the big picture problems that are going on, or what are the statistics. And one of the most important things is that coronary artery disease accounts for 50% of the cardiovascular deaths. So atherosclerosis, the part that I like to study. There are other causes of cardiovascular disease that don't relate to that. But they're just not things that I'm particularly interested in studying. It's not that they're not important. I'm just not an expert in those areas. And so this is kind of a graph of the deaths attributable to cardiovascular disease. And it goes from 1900 to 2008. And what you can see is that it peaked in the 1970s. There were some prominent reports from the Vietnam War and the Korean War, where soldiers were dying in battle, and then they would do autopsies. And when they would go and check their hearts, sure enough, they found that these young, super fit young men actually had very extensive disease. And so that was one of the first indications of how much of an epidemic was on our hand at that time. And so over the years, as we've become more versed about the education of our population, and also have different drugs that affect cardiovascular disease parameters, we've actually been able to reduce the overall death rate from cardiovascular disease. So that's great. And then this is the graph where I said coronary artery disease is 50% of the mortality or deaths from cardiovascular disease. This is the graph that shows that. This is the breakout. And so this is coronary artery disease here is about 50%. Then you have things like heart failure, high blood pressure, other. So that's like a whole list of things that it could be. And obviously stroke is another very prominent cause of death. It's funny because I got 3 screens to look at, so I'm actually confusing myself at some point. So the prevalence of coronary artery disease by age and gender. So men are more susceptible to coronary artery disease, and they're kind of the dark bars here. And then we have age groups across the x-axis, and the y-axis is the percent of the population. And what you can see is when as you age, you get an increased risk of coronary artery disease. And that's always been kind of the way we think about it. Obviously, if anybody's driven by here, the county health building has a little sign out there about February being Heart Month, and in particular this year they're focused on the increased risk of heart disease that happens in women after menopause. So there are some things that we're still working out the mechanisms and causes for. And one thing that I try to remind people of is that it is still the #1 killer in the U.S. is coronary artery disease, and that's what I'm showing right here is the total deaths compared to cancer. So what's a little bit different about cardiovascular disease and cancer is that cancer is there's a little bit more susceptibility or more deaths earlier in life, and that's kind of shown right here. That's kind of in your 50s. And then as you age, your risk of coronary artery disease goes up. So those are kind of some bar graphs and some things that anyone can see at a place like the American Heart Association. This is another graph from the CDC that shows the breakout geographically of coronary artery disease. And so it's pretty amazing if you think about it. Here we are in North Carolina, and there's a bunch of red, and there's a bunch of red down here in other parts of the South. Red is really bad. That's not what we want to see. It's a very high incidence of cardiovascular disease. And in particular, what you can see is that there are areas in the country that are just more susceptible. And part of that is not necessarily genetics. It's probably a lot of it's environment, diet, lifestyle, some other things. And so it'd be nice, and it's a big goal, is to kind of reduce some of these areas that have very high levels of coronary artery disease. And this is one that I always think about, because whether or not you're worried about yourself for coronary artery disease or atherosclerosis, you should be worried about this. And this is the cost in dollars. And this is all forms of cardiovascular disease. And so I just had a little, so that's $1 trillion is what they're estimating in 2030, which is, I don't know, that's a ridiculously large amount of money. So anyway, it's an important public health problem that we have. And we've had it for many years, and we're still working on what are the causes of the disease and what are ways to treat it. And where I work in this field is very far upstream, way far upstream of perhaps things that are going to affect people directly in the next year or two. What I'm interested in finding out is what are novel genes that are going to affect cardiovascular risk. And I'm going to go through some of that in a few minutes. But before I do that, I wanted to just explain what is atherosclerosis. What is this thing I keep on saying? And so it's a common disease that underlies most forms of coronary artery disease. And the definition is just it's hardening of the arteries. So it's kind of a loss of elasticity that happen in the large arteries of the heart or the aorta. And so one of the first descriptions that I could find online was by Leonardo da Vinci. So that's quite a long time ago in the 1500s. And what he described at some point was vessels in the elderly through thickening of the tunics restrict transit of the blood. And basically that was the first description of what is atherosclerosis. I'm going to show you a few pictures of what that looks like. So this is just a cartoon. We're going to go through cartoons first. And so what we have here is, this is your endothelial cells. These are lining your blood vessels. So this here is your artery. This space here, this is where your blood should be. And so in your blood you have LDL, you have HDL. And that's represented up here. And LDL is this big particle. And so what happens, and we don't know all the mechanisms for this and the exact reasons why, but LDL can get caught in the artery wall. And when LDL gets caught in the artery wall, what happens is it gets trapped, it can get oxidized, and there are physical and chemical properties that change to the LDL. At which case, what that causes is an inflammation. And so I'm going to show another picture of that on the next slide here. And so what happens is that LDL gets stuck here in the artery wall. And what happens is your body recognizes this as not good. And so what it says is we need to go clean up some trash. And the trash collectors for your in your body are monocytes, macrophages. So they're cells that your body produces to fight off infection. So it recognizes it kind of like an infection. And they get recruited to the artery wall. And these cells go into your artery wall and they start reacting to this what they're terming an antigen, or not like self. And so what happens here is this is basically what happens when you have any sort of infection. If you cut yourself and you wind up getting that red mark and then there's a little bit of pus and stuff like that, that's basically what's happening on the inside of your artery wall. And this goes on for years, and that can happen episodic. It happens a little bit, then you're fine, then you're not fine. And over years, what will happen is that artery goes from being real thick, or I guess having a really good space for the blood to get through, so it winds up becoming thicker here, and there's less blood flow can get through. And so I've studied this process here where the plaques grow and get more advanced and calcified, and what are the cellular components in there. But ultimately what happens is, at some point, there's a little break in the artery wall. And what happens there is all this stuff that is so necrotic and it's dead cells, it causes the blood to actually just clot around it, and that's ultimately the heart attack. So when people say, I had a heart attack, or I had, the widowmaker, these kind of very acute coronary events, this is what happens where there's a little bit of a break in the artery wall, and a thrombus forms, or a blood clot, and then blood can't get through, and that's what causes the heart attack. So I wanted to just show a couple pictures, because that's a cartoon. So what is this, like, is it real? I don't know. I don't believe cartoons. I have to watch them with my kids a lot, and they're not always, true to form. So this is a coronary artery, and so the real artery should be open about this much. You can see there's all this kind of necrotic stuff in here. Right here, again, this artery should be about that open, and all this stuff in there is occlusion or atherosclerosis. And this one's really bad. So it's almost totally occluded, and there's this nasty stuff in there. And if you're on this side, so that's a normal artery, and this is a normal artery, and this is a normal artery. All the stuff inside, that's the process that we're trying to figure out how that happens. And so this is what we do in science is we'll wind up taking those arteries out of people or our mice or things that we're studying, we'll section them. Basically, we make a candle. So we take them, we put them in paraffin, and then we wind up sectioning them really, really thin. And when we section them really thin, then we can stain them and then characterize what is in the lesions. And that's what this picture is of right here, where this should be the normal artery, and then all this stuff in here is atherosclerosis. This bright red stuff is actually a thrombus that formed. So this is the occlusion that probably killed this person. So that's kind of here. This is their normal artery is here. And this is the disease tissue. So basically, so far, what have we gone through? It's just some basic statistics that say, okay, cardiovascular disease is important, and these are the reasons why. It can actually, as you get older, you may die of it, and number two is going to cost a lot of money, and that there are regional differences in it. And right now, we've gone through some of the really basic mechanisms for what is atherosclerosis, what is coronary artery disease, and we've seen a couple pictures. The other thing that I'm interested in is genetics, and so my actual original interest is in nutrition, and I actually met Dr. Zeisel many years ago. I was a graduate student in the University of Washington, and he came and gave an evaluation for our program. So they have these outside evaluators. And many years later, sure enough, I got to work with him, so I'm really excited about that. But in the intervening years, what I got really interested in was genetics. And so that was a really big change between nutrition and genetics of fields. And so I'm not going to go through all the nuances of genetics, but I'm just going to go through a few little parts about some background information, and then give a couple examples of what I study. And so what is a definition of genetics? It's the study of genes, heredity, and their variation of inherited characteristics. So the last one is probably the easiest to understand. So what geneticists are interested in are things that can be passed between generations. So why am I like my mom? Why am I like my dad? How are things in my family going together? If I have 3 uncles that have diabetes, am I going to get diabetes? That's kind of the one way to think about it. And so as science has progressed, geneticists have become interested in things that cause that. And in particular, the molecular structure and function of genes, the expressions of genes, and the patterns of inheritance. So the expression of genes is one thing that I'm particularly interested in. We're not going to talk about that today, but not only do you have genes that people have heard about in the news, but those genes are expressed. And so that's one of the ways that we must be different. It's not only that our DNA is different, but the patterns of expression are probably different. We'll talk about that in a second. So molecular genetics 101. So phenotypically, we're very different. These are my colleagues at the NRI, and so I kind of took their picture off of our website. And what you can imagine is we all look a little bit different. We're not exactly the same. But genetically, we're almost identical. The estimate is that less than 1% of our genome is variable. So you can imagine that this 1% affects the traits that we can see: our height, our weight, our skin color, our gender. All those things are basically small differences in our genomes. So I just point that out because this is what's important. We're not tremendously different, but these small differences are really, really important, and that's what we're interested in studying and finding what are these small differences and how do they affect cardiovascular disease. So this is Molecular Genetics 101, the second slide. And so one thing that I wanted to show was a couple ideas that if you don't know, I should probably say before we go any further, just to make sure that we're all about on the same page. And that is, number one, our body is, it has tissues in it, and they're made of cells. And these cells contain programmed information, and that programmed information is our DNA. And DNA is organized into genes, and these genes occur on chromosomes. And so here's some pictures of that. So this is DNA right here. So there's a little code of A's, C's, T's, and G's, and they're repeated throughout the genome. And they're organized into genes, and that's this picture right here, this double helix. And those genes are then tightly coiled into something called a chromosome. And then we have many chromosomes in our body, and they're represented here. And so then they're all packed together inside the cell, and that's basically the inner workings of our genetics. But you don't need to know all the details about that. This is what I do. It's pretty simple. So we have people or mice that either have high cholesterol or average cholesterol. So either high or average. And I put this bar to kind of separate them. And so what I'm interested in are these nucleotides, these A's, C's, T's, and G's, and patterns. I'm interested in finding patterns. And so what we have here is this blue nucleotide, and it's different among these different people. So imagine this is all different people. And so there's a small difference here, either G's or C's, but it doesn't seem to go with your cholesterol level. However, if you look at this one here, if you have a T at this position and not an A, you're going to have a difference in cholesterol. That's what I'm interested in. And usually when it's as simple as this, and you can find something that's as simple as this, then even as a scientist, you start to believe it. You're like, all right, well, that seems to make sense. I don't need a lot of hocus pocus to understand. If I have this one difference, I see a difference in plasma cholesterol. So how we get the A's and the T's, it's all the detail stuff that's interesting to me, but probably isn't all that much fun for tonight. So human genetics has really given a lot of information to the study of coronary artery disease, and these studies have found different methods, I guess, to understand cardiovascular disease. One is rare Mendelian forms, and so these are these really, really severe forms of cardiovascular disease that if it was in your family, you would know because you would have many uncles, aunts, brothers, sisters, parents that would be affected. And so that's some of our initial understanding of how plasma lipids play in coronary artery disease is from studies like that. There are these candidate gene studies. So geneticists like myself will make a hunch okay, we think that this gene does this, so maybe if there's a difference in that gene, then it'll affect cardiovascular disease, and that's a candidate gene study. And then more recently, there's been the technologies developed where you can look at everything genome-wide. So you can look at all the differences in a person, and that's some of the equipment that we have at the NRI or they have upstairs here. And that's kind of the next phase of where things are headed. But all that is it's the same thinking about the C's and the T's and high and low cholesterol, but you just can do it with a lot more horsepower, and you can do things a little bit more efficiently that way. But basically, after all these studies have been done, what we've figured out is that there's not one gene. And this is important, there isn't one cardiovascular disease gene. There's many, many small differences between people that affect your risk of a disease, and in something complex like cardiovascular disease. And so I got this from, there's a lipid clinic in Canada that had this on the web, and I thought it was a great example for when genetics is easy. And this is one of these rare Mendelian forms of high cholesterol. So this person's cholesterol, this patient, it's really white and milky. That's not good. So it's white and milky because the plasma is just filled with fat, basically. And then this is what a normal one would look like. So you can imagine that understanding why that would happen is really important to us. And that's what as geneticists, is really, really important, is to understand the phenotype. And that's, for a geneticist, this is the phenotype, hypercholesterolemia. Yep.
What is being shown in these slides, is that the plasma on top?
Oh, yeah. So what happens is they take your blood out and they wind up spinning it down, and then the red blood cells go to the bottom, and then the plasma goes to the top. And the plasma is the yellow part in the normal person, and this white part here is the plasma in the person with a high cholesterol. Okay. And so when we talk about patterns of inheritance, so if you can imagine if that person went to the clinic, they'd start to ask questions about their family. And that's how the initial genetic studies are done, is you try to figure out patterns of inheritance in a family. So we do things like we color people that are affected or sick or died of a disease red, right? So you can imagine that this person here was the first person they can detect that had the disease, and then pass it along to their family members. And that's just the way that some of these diseases work. And so often what we can do then is genotype people, so look for differences in A's and T's and C's and G's, and try to figure out which gene is affecting the disease or the susceptibility to disease. And so that's all human studies. And Steve alluded to earlier that I'm interested in modeling this disease in mice. And so this is a picture of advanced atherosclerosis in a mouse. And so I just want to walk through here that this is the normal artery, and all this stuff in here, that's all the diseased tissue, and the blood can only flow through this little spot. And then what's interesting about these mice is that they're actually getting this hemorrhage here that's forming. And so that's blood pooling in the lesion. It's one of the ways that we think the lesion is broken, and you actually have that thrombus form. And so we can even it's something that's a simpler model, like a mouse, we can model very advanced, very complicated diseases that are relevant to humans. So we talked about this patterns of inheritance. We still do the patterns of inheritance in mice. I just wanted to show this one slide where we take two different strains of mice and we breed them together, and then we breed them together again. So we have two generations, and we just make a whole bunch of mice, and basically we make a family. And we go ahead and we track the genetics through this family that we've created. It's artificial, but that's one of the ways that we do that. And if the mice are bothering you to think about it, think about our original example. So if we had Tim Russert, and we had George Burns. So somebody that's really susceptible and somebody that's really resistant to disease, and we cross them. And then we take their children and we cross them again. We'd be able to then track the patterns of genes that are occurring in each of the children and say, okay, what is the likelihood that one of the kids has a disease? So that's the big picture of what we do.
In your study with the mice, is it typically that they develop the disease because of genetics or because of their outside influences?
So there's, we can do both, actually, is the answer. The very dramatic example I showed you was a mouse that is a genetic cause. And so we can do both, though. And often we like to combine them. Like the real goal is to combine them. You know, say, okay, if you're susceptible, what are things that we change and you're less susceptible? So I'm going to give you just a couple of real research examples. So this is where I hope not to lose you. So our genes are organized on chromosomes. And so this is a picture of chromosome 1, or the results from chromosome 1, for a study that I performed. There's a lot of colors up there. So right here is a likelihood of a gene being associated with atherosclerosis. And so these circles here and these circles here are kind of our genetic hits. And then when we do these studies, we have to take into account how close, how far away do we need to look? Because it's not like the SNP comes up and says, or the difference says, hey, this is me, and the closest thing is going to be it, because it never happens to work that way. We need to study things a little bit more and do some additional experiments. And so we have to look in this window here where it's red. And basically there's a couple genes right here. I'll do it over here. So in this window that's red, this area here, that's where I need to look. And these are the genes that I'm interested in. And so what we do next is go ahead and try to figure out which of those genes are the causal gene. And that's what we're doing in the lab right now. This is just a little reminder to me that this is the building I work in. My lab is basically right here. I'm sure that if you talk to Beverly, you guys can come and check it out sometime. We'd love to have you. We're always interested in kind of sharing what we do. But the NRI is interested in two things in particular: nutrigenomics and then metabolomics. And nutrigenomics is, I'm going to read this to you. So it's the interaction between genes, genetics, and diet, and how they affect human health. So basically, how diet and genes are interacting. And then metabolomics is looking at these small molecule chemicals that our body produces and how changes in our metabolism affect our disease. And we can monitor that by using some special equipment. And so what I wanted to do here was just take a step back and remind myself, because everyone says, diet. Well, it's pretty funny because, diet is, what is diet? It's not a single food. It's everything we eat, the way we eat, when we eat. And so there are many nutrients you can imagine in something as simple as a carrot. And if you eat a salad, there's many more nutrients. And then if you like cheeseburgers like this, what isn't in there? I mean, it's really, you have everything is in there. And there's many chemicals and many nutrients that you can eat. And so there are clear effects of diet on metabolic disease, such as adiposity, plasma lipids. And then what this last little point is, and one of the reasons why I model this in something like a mouse, is diet is heterogeneous, and it's difficult to quantify on a population basis. And there's a couple reasons for that. One is, and probably the biggest one is, people have selective remembering when it comes to what they ate and how much they ate. And so, and I don't know that that's a dishonesty. I think that's maybe just our mind protecting ourselves from ourselves. I don't know. And so one of the things that we can do with our studies in rodents is we can control what they eat, how often they eat, the composition, things like that. And that's one of the reasons why we use them. So here's the second example that I'm going to show. And this is was a project that I started at UCLA, and it's one of the things that I'm excited to continue here at the Nutrition Research Institute is our studies that are focused around a metabolite called trimethylamine N-oxide. And lucky for me, it's a metabolite that relates to some stuff that Dr. Zeisel has studied for many years called choline. And so what we're able to do then at the NRI is combine some of my interest around cardiovascular disease with ongoing knowledge here at the NRI and in Dr. Zeisel's lab about that metabolite and how that might cause disease. So this is example 2. This is brand new research in the last year or two, and this is kind of the cartoon for what we think is happening. And what we think is that there's that our diet is getting metabolized in our gut. And there's this one breakdown of choline in the gut, and that makes something called trimethylamine. And trimethylamine, it smells like rotting fish. And so there's actually a Mendelian disease. So people will have a mutation in a gene, and they can't metabolize TMA, or trimethylamine, to its byproduct, trimethylamine N-oxide. And if they can't do that, their urine and sweat smells like rotting fish. So it's a fairly tragic disease for the people that have it. But because we were able to understand that, we knew, okay, we should look at this gene and see how it relates to cardiovascular disease, or how it relates to trimethylamine N-oxide. And so sure enough, this metabolite, trimethylamine N-oxide, affects atherosclerosis. And so that's really the big part of the study that I was interested in, and how I got very interested. I'm going to go back a slide just to back up for a second. So this metabolite, it's circulating in our blood. We all have it probably. But when we have increasing amounts of it, so this is people without cardiovascular disease, this is people with cardiovascular disease. And so if you have cardiovascular disease, you're going to have more of this metabolite. And so this is really, really interesting, because you can imagine just like a plasma cholesterol test, which will give you a prediction for how likely you are to have coronary artery disease. This might be another measure for people to get. And then this is the other thing that, as somebody that's really interested in vascular biology, that I was really struck by, is that as you have increasing amounts of trimethylamine N-oxide in your blood, you have more affected arteries. So everybody can understand the idea, like a single bypass is maybe not as severe as having quadruple. So, and this number here on the bottom, 0, 1, 2, 3, 4, this is the number of affected arteries right here. So these people are a little bit more sick, is one way to think about it. And so as a geneticist, I'm interested in the structure of genes and how they function. One way they function is they make something called RNA and proteins. And we don't have to go into all the details about that. But what I wanted to show is that once we find a candidate, we go ahead and we follow up. We do experiments to understand how it works. So it's not enough to say that I think a gene is associated with a disease. You need to show that it actually has some sort of function. And so these are some functional studies. And this is like, I guess, studies that are going to really show that if we change the expression of this gene FMO3, that we're going to wind up affecting the levels of trimethylamine N-oxide. And so I just wanted to show, these are studies in mice. We don't do these sorts of studies in people right now. What this is we can take a gene and we can put it in a drug basically and deliver it to the mice and say, okay, if we give you more of this gene, will you wind up having a change in this metabolite? So what we do is we make more of the gene, FMO3, and then if we do that, we make more protein, which is good. Protein is actually the entity that's doing the work in the cells, and then sure enough, we have a little bit of a change in the metabolite. So basically, we're able to kind of tie that together. The other thing that we can do is we can inhibit the gene, the expression of the gene. So what we can do is they're basically small molecules that will block the expression of the gene. And this graph here shows the exact same thing, where if you take this drug and then treat the mice, you inhibit the gene expression, and then sure enough, you'll make the metabolite go down. So I just wanted to give a little bit of a flavor, not so much for how important that particular pathway is, but the fact that when you find a gene as a geneticist, you want to understand how it works, and you do additional experiments to understand it.
Have you studied to see if you can also show a reduction through nutrition or diet?
Of this one, you most certainly can, and one of the ways is there's the precursor, choline. If you change that dramatically, you can do that. There's probably also this idea about the microbiome. So there's a good bacteria in your gut that helps you digest your food. One of the thinkings around this pathway would be that if you modulate that or change the composition of the bacteria in your gut, you might change the amount of this metabolite that's made. Yeah, but so the one thing that I hesitate on is so that's the thinking. I haven't seen anything that like looks like this, like a graph, and I'm like, well, that looks like it makes sense. I haven't seen that yet. So.
Brian, I think what you might not have made perfectly clear is that the TMA is made from choline by gut bacteria. They're the ones who have to be present to make it.
Yes.
That's where that is.
So if you're eating a lot of choline through your eggs or what, is that plus?
I don't know. So I think what's more important there is that there's this complex interaction between your diet, the bacteria in your gut, and your own genetics. And I think it's really much too early to say I should eat more of this or less of this. What I showed is like stuff that's very, very new. So it hasn't really been vetted. And one of the things in science is that's what I think because I did it. So, but it would be really nice to have other people replicate those experiments. And if multiple people can get the same answer, then it starts to have some legs to it. And that's, so the only reason why I hesitate is I wouldn't personally make any changes about that. So, one of the things that I'm also interested in at the NRI, obviously, is studying diet. And so here's just a couple examples. I'll feed different mice or mice that are susceptible to atherosclerosis different diets. And then sure enough, I go ahead and we'll look at different parameters such as plasma lipids or liver gene expression and see how that relates to atherosclerosis. So that's kind of some ongoing work that we're doing at the NRI right now. And so I'm going to stop there. What I wanted to do was just acknowledge the people that have helped me with my research. And I have some members of the lab. This is a picture of us at the NRI. Jody Albright is a relatively local person. He graduated from Rowan Cabarrus Community College in their technical training program. And he works for me. He's the first guy I hired. He's great. And then next to him is Tangi Smallwood. She's a graduate student of mine that actually is up in Chapel Hill. Sammy was a postdoc for me for a short time, but he had to leave because he got another faculty position, so I had to be happy for him. I was sad to see him go, but happy for him. Braden's an undergraduate who works in Chapel Hill, and he lives close by, and over breaks he'll stop and work in the lab a little bit. And then Kristen is a graduate student at UNC Charlotte, and UNC Charlotte's really, really good at bioinformatics. And so she helps me with some of the more sophisticated genetic analysis. And then obviously myself, and then this is Pamela is a technician in the lab who's going to go to graduate school next year, and she'll be surely missed. Other than that, I just wanted to acknowledge some people such as Daniel Pomp and my colleagues at the NRI, and then Stan Hazen at the Cleveland Clinic, who I did this little study with trimethylamine and oxide, and then my colleagues at UCLA that I work closely with. So anyway, thank you, and feel free to ask me any questions.
Does anybody have a question for Dr. Bennett?
This is so.
Yeah, and there is, and so, there's sophisticated imaging techniques, and primarily where that's driven is not so much in not having to cross-section the artery of the mouse, but it would be really, really great from human health is that if there was a way to visualize plaques and know which plaques or which plaques are most susceptible and get those people treated quicker. There is some MRI ways to do that and one of the big things they try to figure out is which contrast agent they can see the plaque better with. But it's still not completely developed. But people are thinking about it. It's a really good idea.
I just wondered if you were aware the government is doing a study like this, and I'm one of 1 million, and you do it through the VA.
Huh.
And it's really big.
Yeah, I don't know about that so much, no. But I kind of wish I did.
But what they're doing is just through the blood, the DNA and stuff.
Yeah. And that's one of the things, I think one of the things that geneticists do that work mainly with humans is they do studies just like you're describing, where you wind up, you collect populations of people, and you have a doctor's exam or a nurse's exam, and in part of that exam, they'll take your blood, and from your blood, they can isolate DNA, and what they do is they store that DNA, and they'll process it, store it, and when they get a batch of it, they wind up doing some assays, or they genotype the people to figure out what polymorphisms you have versus other people. And so that's a fairly common approach.
I just thought it might be, maybe check sometime and just talk with them.
No, actually, so that's one of the things that's actually been very good about science is that all the data on studies that you're referencing is actually deposited in repositories. And one of the things that we do is we share our data because there's no reason why a result I have is really mine. It's not really mine. Most of the research is supported by tax dollars. So we share those things. And so I appreciate your comment because it's an important one. I think just systematically we're really invested in understanding other people's results and how they apply to ours, so we can figure out better therapies for people. So.
If they get 1 million people, I'm one of 1 million. That's a lot.
That is a lot of people, and you're right. And the most important thing that you just said is a lot, because for genetics, more is better. And so I think you understand genetics perfectly. So, and I hesitated actually to put this in, but maybe I should have. Is that a lot of the research that we know, or a lot of what we know about heart disease, came out of a study called the Framingham Heart Study. And so what they basically did is they went to Framingham, Massachusetts, and they studied a bunch of people there, and then they studied their offspring. And really what you need there is you need the follow-up. You need to study people over time, because we're all alive today and healthy if you enter the study. They want to know what happens in 5 years, and it's really important that that follow-up time point is really, really helpful for scientists.